Welcome to my channel Learnomate. In this video, we will going to discuss the purine synthesis pathway. Also, we will going to discuss the salvage pathway in the next part of this video, in which we will going to discuss the disorders because of the excessive purine synthesis. So starting from the first step of the purine synthesis. This is the structure of a purine ring. A purine is a nucleotide that is important for the synthesis of inosine monophosphate and adenine monophosphate because the purine contains the two nucleotides, the adenine and the guanine. So the purine ring is formed from the different amino acids and the carbon skeleton. The nitrogen and the carbon skeleton is formed because of the presence of three major compounds including the glycine, the aspartate and the glutamine. These three amino acids provide the nitrogen atom to the purine ring, while the carbon atom are coming from the two compounds, that is the carbon dioxide, and the second one is N10-formyl tetrahydrofolate. These two required for the carbon ring synthesis and the nitrogen coming from the three amino acids. These all combined to form a purine ring. So this purine ring is going to synthesize into different compounds, that is the inosine monophosphate and the adenine monophosphate, that is required for the major functions of the body. So first step for the purine synthesis is here the conversion of the ribose 5-phosphate into the 5-phosphoribosyl 1-pyrophosphate, also called as Prpp. This product is the initiator of all the cycle of the purine ring. This ribose 5-phosphate is coming from the glucose 6-phosphate. The glucose 6-phosphate through an HMP shunt convert into the ribose 5-phosphate, and the ribose 5-phosphate plus the ATP. These two are the precursors for the purine synthesis. This purine synthesis occur major in the liver. So in the liver, these two are the precursors, and when the ribose 5-phosphate and the ATP combine with the presence of the pyrophosphate phosphoribosyl pyrophosphate synthetase enzyme, there will be a production of the Prpp. This Prpp compound is an important compound, as I have told you that this is an important initiator of all the cycle of the purine synthesis. So the enzyme required the conversion of ATP into the AMP and also the magnesium as a coenzyme. The activators of this step is the phosphate, and inhibitors are the purine ribonucleotides. This step is a rate limiting step because it is very important and the first step of the purine synthesis. Now this Prpp, that is the 5-phosphoribosyl 1-pyrophosphate, is going to convert into the second compound, that is the 5-phosphoribosyl amine. This 5-phosphoribosyl amine is going to require the nitrogen atom, and that nitrogen atom is contributed from the glutamine. This glutamine is going to form the nitrogen compound of the purine ring, and there will be a formation of 5-phosphoribosyl amine. This step is also under the control of activators and inhibitors. So the activator is 5-phosphoribosyl pyrophosphate, that is the compound that is produced. If it is in excess, there will be production of more and more 5-phosphoribosyl amine. If it is negative, then there will be less production, and the negative inhibitors are AMP and GMP. Now this compound getting another nitrogen atom from the glycine is going to convert into the GAR. This compound now going to convert into the next compound with the combination of N10-formyl tetrahydrofolate, and we have remembered that this N10 tetrahydrofolate will going to combine and contribute the carbon atom. So now there will be an addition of a carbon atom in the ring. The carbon atom has been added in the next step, and now this compound is going to convert into the next compound with the addition of again a nitrogen through the glutamate amino acid. Now this nitrogen will going to combine, and there will be a production of a compound called N-formyl glycimidine ribotide, FGAM. This compound having a 1 carbon atom and 4 nitrogen atom. Now this carbon atom and nitrogen atom in this compound will going to convert in the presence of AIR synthetase into the AIR compound, and this AIR, that is 5-imidazole ribotide, is having the nitrogen plus the carbon atoms, and this is going to form a ring. Because the purine is a double ring structure, so one ring is formed here in the halfway of this cycle. Now, after the conversion of different steps with the combination of the carbon atom from the N10 formaldehyde tetrahydrofolate and nitrogen from the aspartate, there will be a production at the end of this cycle. There will be a production of 5-imidazole 4-carboxamide ribotate, and this compound is now going to convert into the major one, that is the FAICAR, and this FAICAR compound is a conversion because of the presence of N10 formaldehyde tetrahydrofolate, and this N10 formaldehyde tetrahydrofolate is going to contribute on 1 carbon atom more. So at the end, what will happen with the presence of cyclohydrolase enzyme and IMP cyclohydrolase, we're going to convert this compound into the inosine 5 monophosphate. And this is your end product that will going to form into the different products, that is adenine or the guanine monophosphate. So there is a 2-carbon ring has been formed, which having the nitrogen and the carbon atom contributed from the different amino acids and carbon dioxide and N10 formaldehyde tetrahydrofolate. Now up to this, the cycle is under the inhibition and excitation. The activators are the phosphoribosyl pyrophosphate and the inhibitors are different compound, plus the methotrexate in the human being is an inhibitor of tetrahydrofolate reductase. So now what happens, this inosine will going to convert into 2 pathways. Now guys, this inosine monophosphate can go to convert into the adenine monophosphate or into the guanosine monophosphate. If it will going to convert into guanosine monophosphate, it will first form the xanthosine monophosphate. The xanthosine monophosphate is formed under the preparation of inosine monophosphate dehydrogenase enzyme. And this xanthosine monophosphate is an intermediate compound to be converted into the guanosine monophosphate that required the importance of a glutamine as an amino acid and the ATP. These two precursors, the glutamine and the ATP, are must for the synthesis of the guanosine monophosphate at the end of the cycle. But if the inosine monophosphate will go to the another pathway, that is the formation of adenosine monophosphate, this adenosine monophosphate under the influence of enzyme adenosine succinate synthetase will going to convert into the adenosyl succinate. This adenosyl succinate under the influence of the adenosyl succinate lyase will going to convert into the adenine monophosphate. But here important, the 2 precursors for the conversion of adenine monophosphate from the inosine monophosphate is the aspartate and the GTP. The GTP and the aspartate are must for the formation of adenosine monophosphate, similar to the guanosine monophosphate. Now these 2 compounds are the precursor for most of the nitrogenous bases for most of the compounds, because these are the monophosphate. They will going to convert into the di or the triphosphate under the influence of glucokinases and nucleokinases. So different compounds will be formed, and that is the end of the purine cycle. Now if the purine cycle has been formed and different rings of the purine have been formed, there will be a excretion or the salvage. The purine salvage pathway will be discussed in the next part of video. Thank you for watching.